Hi, good afternoon. Welcome to the Patient Engagement and Cancer Genome Sequencing Pre-Submission webinar. My name is Liz Glanders, and I'm happy to present today on behalf of our team. This includes Dr. Leah Mechanic and Sylvia Cho, who are also here with me and will be available for questions. So just starting with a few webinar logistics, all of the lines will be in listen-only mode, but you can submit questions at any time using the Q&A or chat panel and select all panelists. So you may need to actually activate the appropriate box in order to do this, and you can do that by using the floating navigation panel found on the center of your screen and shown here. Closed captioning is available by clicking on the link in the chat box, and just to let everyone know that this webinar is being recorded. So this is our second pre-submission webinar for these FOAs. The first webinar was held on August 7th, and you can find slides from that webinar as well as a list of frequently asked questions and other program details on our EGRP website, and the link is provided here. You can also find get access to the recording of our first pre-submission webinar using this the following link. And today we're going to be talking primarily focused on the frequently asked questions we've received in regards to both of the FOAs. So our goal is to keep the discussion and our responses general and broadly applicable to all applicants or teams. So just a quick outline of today's webinar. I'm going to start with a very quick overview of both RFAs and then highlight some of the frequently asked questions that we've received. Then at the end of the webinar, we will have time for additional questions to be taken from the audience. Okay. So the patient engagement and cancer genome sequencing RFAs are associated with the Cancer Moonshot initiative and fall under the scientific priority area designated as Recommendation A to establish a network for direct patient engagement. And the National Cancer Institute intends to support this network, which will include several U2C research centers, which will be supported under the RFA-CA-19-045, as well as one U24 coordinating center, which will be supported under RFA-CA-19-046. And it's our vision that the patient engagement and cancer genome sequencing network will function as a collaborative network, allowing for the research centers to address common issues, share best practices and lessons learned, and utilize common methods where appropriate. So the overall purpose of the PE-CGS network is to promote and support direct patient engagement of cancer patients and post-treatment cancer survivors as participants in the cancer research enterprise, and to use direct patient engagement approaches to support rigorous cancer genome sequencing programs addressing important knowledge gaps in the genomic characterization of tumors in areas such as but not limited to rare cancers or rare cancer subsets, highly lethal cancers, cancers with an early age of onset, cancers with high disparities in incidence or mortality, or cancers in understudied populations. So I would just emphasize that the first goal must fulfill the goals of the second, which is specifically to address knowledge gaps in the genomic characterization of a particular cancer, a particular type of cancer or tumor. So this slide shows the key details for RFA-CA-19-045. The earliest submission date for this RFA is September 30th, and we are asking for letters of intent, which should be submitted 30 days prior to the due date. So that would be September 30th, 2019. There are two application due dates for this RFA, October 30th and July 30th, and NCI intends to commit $12 million in total costs in FY 2020 to fund up to 3 awards. Application budgets are limited to no more than $2.5 million per year in direct costs and should reflect the actual costs of the proposed program. And the maximum period, project period for these awards is 5 years. So here's are the key details for RFA-19-046 for the PE-CGS Coordinating Center. It has the same earliest submission date, the same date for a letter of intent, the same due date for the letter of intent. And these letters of intent should be sent to the email address that's shown on the screen here. It has the same application due dates. However, it's important to mention that if an applicant is selected after the first round, this FOA will be expired. So NCI intends to commit $500,000 total costs in FY 2020 to fund a single award. The application budgets are limited to no more than $350,000 direct costs, and again should reflect the actual needs of the proposed project. And again, the maximum project period for this RFA is 5 years. So moving on to the frequently asked questions, we've tried to group these by the particular content area in which they fall. So starting with frequently asked questions regarding the application details, several people asked us what was required for the letter of intent. So this letter should include the following information. There should be a descriptive title of the proposed activity. You should include the name, address, and telephone number for all of the PIs, the names of other key personnel, participating institutions, and as well as the number and title of the relevant funding opportunity. So whether it's in response to 19-045 or 19-046. And again, these LOIs should be submitted to the following email address. So we had several questions about RFA 19-045 from investigators who wanted to know if they could not complete an application in time for the October 30th submission deadline. So there are currently 2 receipt dates for the FOAs, October 30th, 2019 and July 30th, 2020. And this is to allow for either resubmissions or for new applications that missed the initial submission deadline. However, it's important to recognize that this second date will depend on the availability of funds. So continuing with application details, investigators asked whether it's possible to apply to both the U2C and the U24, so the Research Center and the Coordinating Center RFAs, and the answer to that question is yes. The same institutions or investigators can apply to both. Others asked, what is the distinction between the participant engagement unit, so Unit 1, and the engagement optimization unit, Unit 3? So the participant engagement unit, Unit 1, is responsible for the implementation and operations of patient engagement, while the engagement optimization unit will provide supporting behavioral or communications research to develop and test the optimal approaches to patient engagement. So in other words, the participant engagement unit should develop the initial protocol based on the current state of the science, but allow for flexibility in the protocol to adapt according to the research findings from the engagement optimization unit. So in other words, the research performed in the engagement optimization unit should iteratively guide the implementation and operations of the participant engagement unit. Okay, so continuing with application details, the last question we had in this category was, is there an extension to the submission deadline for investigators participating on study sections? So the answer to this question is no, that since these RFAs do not use the standard receipt dates, we're unable to use the continuous submission policy. In addition, these FOAs will not accept any late applications, so there's no extension to the submission deadline for members of study section, unfortunately. Okay, so moving on to questions related to the study design. We had several investigators ask about the appropriateness of a particular cancer type for this FOA, for the research centers RFA. So the U2C research center should be centered on addressing a unique research knowledge gap in the genomic characterization of tumors. And knowledge gaps proposed for characterizations are expected to be mainly identified in the interest areas, the 5 interest areas listed below, which I highlighted earlier. However, we want to be sure it's clear that knowledge gaps outside of those first 5 interest areas may be proposed along with a strong scientific justification. Another question we received was specifically about interest area 3, which was cancers with an early age of onset. And investigators asked whether this referred to pediatric cancers or cancers that traditionally occur in older individuals but have been shown to impact people of younger ages. So for this interest area, We were indeed thinking about the latter, so cancers that typically affect older individuals occurring in participants of younger ages. However, it should be clear that many pediatric cancers are rare and or understudied, so these cancer types could also fit under other areas of interest. Another question that several folks posed regarding the study design was how many participants or tumors are required. And so here we'd say the proposed minimum number of unique tumors to characterize, or in other words, the proposed number of participants to engage should be sufficiently high to ensure rigorous and interpretable results for the specific research that's being proposed in the application. So this number could vary depending on the cancer type being selected. Others asked, specifically, we have already identified the population we want to engage for participation. How important is it that we address a research gap in the genomic characterization of tumors? So for applications to be considered responsive to this RFA, it's critical that the team identifies and articulates the particular knowledge gap in the molecular characterization of the particular cancer or population subset that will be studied. So it's the application really should be driven by the knowledge gap that could be filled with the successful completion of the project, and it's important to provide a strong justification for the chosen knowledge gap and selection of participants. Others asked us whether it was possible to use existing infrastructure or resources for enrolling cancer patients or participants. And we responded that investigators are encouraged to build on existing infrastructure, but it should also be possible for any participant meeting the inclusion criteria to have the ability to enroll in the project. So the last question regarding the study design was how much preliminary data is expected for the engagement optimization unit, or unit 3. So applicants should provide some detail on the questions that they anticipate will be the major relevant questions to begin with, and include any specific pilot project ideas. So, for example, this could be testing different modes of obtaining informed consent, or testing various methods of gauging participant preferences in terms of return of results. And the applicants should provide enough details for reviewers to feel confident that the research will be feasible and successful and will inform the larger protocol in Unit 1. However, by design, the engagement optimization unit will be, their work should be iterative in nature, so providing comprehensive details of all the work that will be done or completed in this unit would not be expected. So we had several questions regarding the expectations for CLIA certification. So the first one was, should all of the required genomic characterizations, the whole exome sequencing, the RNA sequencing, and the low-pass whole genome sequencing be performed in a CLIA-certified lab. And we did clarify that the whole exome sequencing should be performed in a CLIA-certified lab, allowing for the return of individual-level genetic information to those participants who are interested in receiving such data. However, the low-pass whole genome sequencing and the RNA sequencing do not need to be done using a CLIA pipeline. Another question we received was, is it possible to perform the whole exome sequencing in a research lab and follow up with clinical validation in a CLIA-certified lab? And so we clarified that hybrid approaches such as this one, sequencing in a research lab and validating of actionable targets in CLIA would not be appropriate. We're expecting that the policy around returning information to participants will evolve, and so having the whole exome sequencing performed in a CLIA environment will give us more flexibility for the policy to evolve and allow for broader return of results if so desired by participants. Okay, so the last question we had was several people asked whether there's any flexibility in the selection of platforms for the genomic characterization. And so we would just like to reiterate that for each cancer or population subset, the characterizations must include, at a minimum, the whole exome sequencing, the low-pass whole genome sequencing, and the RNA sequencing. However, applicants can propose additional types of genomic characterization if appropriate for the specific cancer being studied. Applicants will need to submit or include a strong justification for these additional characterizations, and they should all, or they must all, be included in the allowable budget, so no more than $2.5 million direct costs per year. So that summarizes the primary questions that we've been receiving, and just as we're waiting for questions to come in from the audience, I'll just repeat a couple of things. So there is, so this is where at any time you can submit questions to us about the various FOAs.
Okay, so just as a reminder, as you're submitting questions, that we're going to be doing more general questions, and if you have very specific questions about your aims, please email us. So, okay, so the first question that I'll ask that came in is that whether there is an expectation that the optimization unit will be the evaluation arm of the center, or in other words, are you expecting to see a formal program evaluation and plan for the different units of the center?
So I think this actually, the other point that I should have made in addition to where you can submit these, you can submit questions to us is that on the EGRP webpage, I mentioned that we have a list of frequently asked questions, which we didn't have a chance to address all of them today, and I think this one is actually there as well. So we are expecting that the evaluation, the engagement optimization unit will provide and will conduct an evaluation of the various methods that they're testing in Unit 3, but not necessarily that they would provide, serve as the evaluation center for the entire application, because it may not be appropriate for them to, for example, conduct the evaluation of the genome characterization unit.
And then just to add that each one of the units does have a section on benchmarks and progress that you have to include within the research plan.
Great.
Okay. So another question was whether you could comment on the reporting requirements for germline data.
So I'm assuming this is asking about what we're expecting people to return to participants. And so I think we specifically asked, we anticipated that we'd start by asking applicants to return the pathogenic variants for the cancer genes in the ACMG 59 panel. But this is something that that could evolve across the course of the project period.
And based on the evaluation of participant preferences. So I can do this question. Okay, so we had a question about an IDE requirement for the RFA-19-045, and this is something that, number one, I encourage you to reach out to us by email to discuss this question because it's very specific. But we require that the investigators talk to their institutional IRBs about the possible need for an IDE, and then document the outcome of those discussions, and then have the necessary pre-submission discussions about this. So this is something that you know, will depend on your IRB, but I think if you have a specific question about this, I encourage you to email us about this question. So there was a question about the, so the next question that came in is about where the main research component is, whether or not the main research component is housed in the engagement optimization unit, where there may be trials conducted.
So I guess I would just clarify that the that each of the units will have their own set of specific aims, and that it's true that the engagement optimization unit will likely have research questions related to the optimization of the patient engagement methods, and how to best communicate with patients, engage patients, but all of the research units should have a set of specific aims.
Okay, so I think that we had another question about the genome characterization and participant engagement unit, so I think this overlaps with the question that you just answered. I think, so one question we had was about, you know, and you just mentioned whether or not they have specific names. Can we have multiple PI applications?
Yep, so the answer to that is yes.
So I'm looking, we're waiting for, we're having some questions come in. So I think we've addressed.
Yeah, so I would say what Leah mentioned is important to that you can always reach out to us with specific questions, and we will be happy to respond as they come up.
So the next question was about the letter of intent. Is this mandatory?
Yeah, so the letter of intent is not mandatory, but it's extremely helpful for us if you do submit a letter of intent, because it will allow us to start thinking about potential review panels and what review needs that we might have. So technically it's not mandatory, but strongly encouraged.
So what about sample size and power for each unit? Is that something that's required?
Well, I think that I mentioned that, so we're not, let's see, I think that was, yeah, so in this question, we talked about that. We're not requiring a specific number for sample size, and this number should depend on the specific research that's being proposed. But it's important to justify that your proposed sample size would ensure, you know, a rigorous and interpretable results at the end of the completion of the project.
And I guess the one thing I'll add is that for Unit 3, because you may be conducting some behavioral research studies that are outside of they're not your sample size for the genomic characterization, but there may be sample size for evaluating consent or something. So you may have requirements for sample size within that Unit 3,
yeah.
Your research questions.
Yeah, that's right. That's a good point, Leah.
So there's a question about that came in about, can we talk about the evaluation component of the engagement optimization unit? But I'm not totally clear about what that question means. So whoever asked that can kind of provide more information.
Yeah, sure. I think you can get more information, but I would imagine that we are expecting the applicants to articulate how they would evaluate each of those iterative questions that's being proposed.
Okay. So there's a question about whether or not there's an expectation that application should outline how data will be coordinated with the center and cross centers in the network.
I would think that that they should articulate certainly how the data would be coordinated within the center, and then also how they could be coordinated and used collectively across the entire network. So I would say that, yes, there is that expectation.
Yeah, we do expect within the applications for people to kind of talk about how they can collaborate across the network, how they may adapt to common measures that the network may describe, and also how they would put the data into the genomic data commons. You know, as far as
yeah.
So that is.
At the end of the completion of the project.
At the completion of the project.
So one thing to maybe mention, Leah, is that we that there is a portion of the budget in the U2C research centers, which is starting in year 2, expected to be set aside for collaborative projects across the network. And so this is where we see people being able to propose studies that would address sort of common issues that are challenging several of the U2C research centers. So there was a question about doesn't seem to be a separate data analysis unit, and should this component be included within the genome characterization unit? And that is, yes, where we expected that information to be included.
Yeah, and I think we have outlined sort of staff requirements in the genomic characterization unit to address those informatics needs. Another question came out about with the RNA-seq and whole genome sequencing being performed outside of a CLIA environment, can you comment about providing this data back to patients?
So I think that would need to be a discussion that applicants would have with their IRBs, but our understanding is that it really does need to be performed within a CLIA environment in order to for the data to be returned back to patients. And but I will note that this is a field that's changing quite rapidly, and so that could change within the next few years. It could be that the NIH develops a framework through which research data could be returned to individual patients. But until something like that were more developed, we would stick with the guidance of only returning CLIA-generated data.
So there's a question about the engagement optimization unit versus the participant engagement unit. Is it correct to say that the engagement optimization unit does the developmental work and the participant unit carries it out?
Yeah, so that the way that I tried to describe it earlier was to say that Unit 1 sort of develops, well, the entire team would develop a initial protocol in which Unit 1 would implement, and that then Unit 3 is trying to iteratively test better methods to engage patients, and that Unit 1 would then update their protocol based on the results from Unit 3.
And I think,
Sylvia, did you want to also add something about that?
Sure. Yes. If you think about the sort of behavioral science and communication expertise that's in your team, I think that group would lead the charge in terms of outlining how to ask the right questions, how to measure, and how to inform the protocol, which is in the Unit 1. So in the way I think about it, sort of the overall protocol happens in Unit 1. That's kind of the more official, I guess, route of, engaging, collecting data, or getting consent, collecting data, and so forth. And there may be some pilot studies and other type of ways to know, how do we do this consent process? How do we find the right participants to engage? That may happen in Unit 3, which then feeds into how Unit 1 is carried out. I don't know if that adds a little more, but
yeah, thank you. Yeah, thanks, Sylvia. So I mentioned the collaborative set-aside, which started a little flurry of questions coming in, and so in particular, how applicants would be expected to how much detail is expected to be included about those potential collaborative projects. So I think in the application, individual applicants could anticipate what some ideas around the issues that are likely to be challenging to all applicants. So, for example, medical record data abstraction. And so you could propose some ideas about anticipated questions that could be addressed and how you are poised to collaborate. But we do anticipate that, especially since these projects won't start until year 2, that it will really be a collective conversation around what projects make the most sense. So I guess the applications, you'll be able to show that you're tuned in to what questions will likely be most relevant, and how you're uniquely poised to collaborate with other funded groups.
Right. Okay, so we have a follow-up from that engagement optimization unit question. Then they basically, excuse me, wanted to know how detailed and if the evaluation plan should only cover Unit 3 activities or also about Unit 1, but it seems like only Unit 3, so correct it if I'm wrong. So this once again gets at that question of evaluation.
So I think that what Leah mentioned earlier is that each unit has, and if you refer back to the RFA, there's specific details around evaluation criteria within each unit. And so we really aren't expecting Unit 3 to be performing evaluations for Unit 1 and Unit 2. Leah, did you want to quick comment on that, or maybe clarify?
Yeah, so I think Sylvia had added to this discussion too, is that Unit 3 is really looking at the overall sort of at our engagement, but each unit has its own evaluation and benchmarks and plans to address any problems in reaching those benchmarks.
Yeah, okay.
So there's another question that came in about the populations for the genomic characterization. Should all the populations be US-based, or can groups in underdeveloped countries also be included for the enrollment or profiling?
So I think that including underdeveloped countries for enrollment or profiling would be okay. There is a limitation, and Leah, you can remind me, but I think the only limitation in terms of the submitting institution needs to be a US-based institution, but there is not a limit on the populations that could potentially be included.
Right, so the FOA does allow for foreign components, but foreign institutions cannot apply. And if anyone has any specific questions about countries that they might want to include outside of the US, I encourage you to email us, and then we can just follow up with you independently. So I don't have any more questions coming in right now. We'll give it maybe another minute to see if we have any more burning questions. Oh, what's the, oh, okay.
Maybe one other thing that I would, that I mentioned earlier, but I would just clarify is around the 2 receipt dates and especially since I misspoke earlier about the amount of money we have set aside. So there is, NCI does have, for the U2C research centers, has currently committed $12 million in total costs in FY 2020 to support that RFA, and we do intend to ask for additional funds to support additional applications, but at this point, that will just depend on future budgets and future availability of funds.
Okay, so another question came in about whether the engagement and optimization units can be led by the same people.
So that's Unit 1 and Unit 3. Sure, they could be.
And I guess just making sure that they have adequate expertise to lead these two units, in particular, the communication and engagement expertise.
Maybe one last thing, Leah, if we don't get any additional questions, I feel like this came up in several of our calls. So just to emphasize that we really do see those two components of the participant engagement and the genomic characterization, those areas of expertise really should be, are both important and equally important, and we are not, we're committed to having the appropriate expertise and focus on both of those areas.
Okay, so, oh, there's more? I thought they were done. Okay, so, I don't see any more questions.
Yeah, so I guess, go ahead, Leah.
Yeah, just, so I guess since we're not having more questions come in, but please feel free to email us at the NCI-PE_CGS email address. If you have any questions, we are updating our FAQs on the website regularly, and we'll be following up with an update this week to add more content, as Liz had described in this webinar. So, you know, we're happy to address any questions that you may have.
I just want to clarify, so that's an underscore, NCI_PE-CGS, because later when it's underlined, it's hard to see that.
Hard to tell that it's an underscore.
Yeah.
But, and you can, yeah, so we'll have all this information posted on our website.
Okay, thank you very much for joining us, and feel free to be in touch if you have additional questions.